So good morning, everyone. I'm Danielle. I'm a postdoc at Yosef Barash's lab at University of Pennsylvania. And I'd like to thank the organizers for selecting my talk and putting me when you guys are all very awake and can hear how exciting uORFs can be. So I will tell you about how we explore the landscape of regulatory uORFs in BMPR2 and their potential as therapeutic targets. So what are uORFs? They are open reading frames that reside in the 5' UTR of untranslated regions of the mature mRNA transcript. They have a start codon occurring before the protein start codon, and an in-frame stop that can occur either before or after the protein start codon. So why are they important? Because when a ribosomal subunit begins scanning the mRNA transcript, it will encounter the uORF start codon. And then it needs to decide whether it will recruit the 6DS large subunit and begin translating the uORF, or continue scanning the uORF by itself until it gets to the protein start, or dissociate from the mRNA altogether. In this manner, the uORFs can modulate translation of the protein that is downstream on the same transcript. They can repress the translation by any of these means, or induce it by these. Previous work from our lab showed that uORF start and stop codons are under strong negative selection. We can look at MAP scores, mutability adjusted proportion of singletons, which is a measure of the relative abundance of singletons in a data set. The higher the MAP score, the stronger the negative selection is acting on the feature that we are looking at. So this is used as a measure for conservation. And we can see that our start codons have a very strong selection on them if we consider them as a non-coding region. And you can see that at the panel on the left. Specifically, disrupting start codons of short uORFs will have a higher MAP score greater than missense mutation. The stop codons displayed on the right panel have a very even stronger MAP scores than missense, and they are even approaching the predicted loss of function in variants that strengthen the stop codon. So this is showing us where the negative selection is at its strongest. So hopefully I've convinced you that uORFs are very important and we need to study them. And now I would like to present our workflow for variant interpretability and functional testing. By mapping and annotating uORFs, we can inform refine our candidate selection for experimental validation and potential therapeutic development. So for this, we compiled an existing database of all experimentally detected uORFs called RiboUORF. The actively translated uORFs can be detected by a method called ribosome profiling, which sequences fragments of RNA that are actively translated at the time that we started working. This method is currently still very limited to cell culture because of the amount of cells and the different manipulations needed to be done to them. And as so, it is also limited to the conditions that it is testing at the moment, just like all RNA sequencing methods. So for our task, we developed an algorithm that predicts the uORFs from the entire human transcriptome. And our combined data set includes different features and parameters.
Danielle, here you are, and it should be a 5.
Yeah.
This is where you.
It's okay. I'm not gonna really restart. It's all good.
Yes.
Thank you.
Okay, so, all right. So when we compare them to the wild type, 4 uORFs upregulate and 1 uORF downregulates the reporter protein translation. So this was very excited, and we said, all right, we want to look at therapeutic targets. So we turned to antisense oligonucleotides. So ASOs are short single-stranded nucleotides that are modified, and they target specific RNA sequences and physically block the ribosomes or RNA binding proteins from attaching to it. So we designed ASOs to treat the cell line that when induced, it expresses a luciferase reporter protein that is activated by the BMP pathway. This system enables us to measure the endogenous protein response to BMPR2 upregulation. And indeed, we see that when we treat the cells with those ASOs, we see increased reporter activity, indicating that treating the cells with it leads to the activation. Treating the cells with it leads to the activation of BMPR2, which in turn activates the entire BMP pathway. We were very encouraged by these results, so we turned to patient cells, pulmonary arteries that were taken out of patients undergoing lung transplant, and of course, pulmonary arterial hypertension patients. And we treated their cells with the same ASO, and we saw that they have indeed an increased amount of protein. So we didn't stop there. We designed a mouse-specific ASO, which targets the same regulatory uORF in the mouse genome, and injected it into wild-type mice through their tail veins. We used ELISA, which is a colorimetric protein quantification method, to quantify the amount of BMPR2 protein in the pulmonary artery versus other tissues in the mice. And we saw a very tissue-specific response to the ASO treatment in mice that were treated with ASO versus different controls. So this by far is our most exciting result, and we're now wrapping it all up to a paper. So to summarize, we have created a database that includes both experimentally and computationally detected uORFs. And our database includes annotations and parameters that improve our selection of candidate uORFs for functional testing and aid in variant interpretability. The tested uORFs can be strong modulators of translation of their downstream protein. And we can design ASOs that target those functional start and stop codons of uORFs as a means to modulate translation both in cells and in mice. With that, I would like to thank my mentor, Youssef Barash, and our collaborator, Nick Hand, and all my co-authors and my colleagues in the lab. Thank you for listening, and I'm happy to take questions.
So maybe while we set the online speaker, we could take a question. Can you help setting up the online speaker? Okay. And so do we have a question?
Can I ask?
Yes.
Go to the mic.
That would be great.
Okay. So as a computational person, I'm interested in high-throughput data set. I think there's a way to turn your discounter assay to test many thousands of uORFs in parallel or variants in uORFs, so we can get data from machine learning.
Yeah, thank you for the question. Yes, definitely, and it's in the works.
Is it?
It's in the works, maybe next
Okay.
ISMB.
So I'll be looking forward to see the data.